I'm happy to go over a little bit about the vaccine for COVID. It's been in the news the last few weeks, and in particular this week. I don't know if anybody here, is it approved as of today by the EUA? I don't know. Hadn't heard. But I gave grand rounds yesterday on the vaccine, and I put together a toned down version of what I presented to the physicians, and I thought it would be useful to present it here and maybe put it up on the web. So with that, we'll get started. I titled the talk, SARS-CoV-2 Vaccine, a Light at the End of the Tunnel. I certainly hope it is. And we'll go through some of the discussion points. First thing is just to review the basic concept of vaccination. We'll discuss the different vaccine types that are being developed for COVID-19. We'll describe the benefits of the vaccine and talk about some of the processes and challenges of the national vaccine effort. And then I'll go through a brief listing of questions. I get a lot of questions, and see if I don't touch on things that most everybody has on their mind. So with that, keep going. I think first, just from the perspective of, why would this be something you'd want to do? And I think that there are basically 4 points to think about. One, obviously for protecting yourself, but as well as your family, keeping your residents, the people around you, safe, helping stop the spread within the community and set the example for others. I think as a infectious disease organization focused on public health and infection, we really are a leadership organization for this, and we want to set the example for others. So I think that would be important as well. So let's think a little bit about vaccine science. What does it mean to be vaccinated, and what is our goal of getting vaccinated? Well, first we got to talk a little bit about what vaccination is. So just to briefly summarize or talk about that, vaccination means it's a way that we give what's called an antigen to stimulate the immune response through active immunization. So when you get a shot, that's an immunization. When you're given a shot, you're given an antigen, and we hope that that's going to produce a reaction that'll protect you for an infection in the future. A vaccine is what we call an immunobiologic. Immuno meaning stimulating the immune system, biologic meaning that it's from the biological system. So an immunobiologic substance designed to produce a specific protection against a given disease. So in this particular case, we're talking about an immunobiologic based off of SARS-CoV-2, that's the virus that causes COVID. And we hope that by giving that to each of us, it'll protect us against COVID. That's the principle. A vaccine is an antigen, or it's antigenic, but it's not pathogenic. That means that it wouldn't be good to give us a vaccine that causes disease. So getting a vaccine provides a stimulus to the immune system through the antigen, but it is not going to make you sick, meaning it's not pathogenic. So that's the concept of a vaccine. When vaccinated humans, we all produce antibodies called immune globulins, and we produce what's called T cells that kill the virus. We call this cellular immunity. So there's 2 parts to the immune system. One is the antibody part. We also call that the humoral system. And the other is the cellular system. That's known as the T cells. Here's a schematic of the concept of an antigen and what it looks like. So I gotta minimize that. So number 3, if you will, in the context of thinking about COVID, COVID is caused by a virus called SARS-CoV-2. Number 3 would be the SARS-CoV-2 virus. Number 2 would be a structure on the surface of the virus, as would number 4. And those structures on the surface of the virus would be called antigens. The idea would be to somehow get the body to recognize the antigen without getting sick from the virus so that it produces what's called an antibody. The antibody is represented here by this pink Y. And you can see that schematically, it's a reasonable representation. This antibody matches this antigen. And when that happens, it can bind to the virus should we get exposed and prevent that from getting us sick. So again, that's the concept of vaccination using an antigen against a pathogen. The antigen would be separate from the virus. So how do we measure the effectiveness of a vaccine? There's two different ways that we do that. There are indirect measures, it's one way, and then there are direct measures, and we'll talk a little bit about both. Indirect measures, I already alluded to with the prior slides. There are three principal things that we look at. One is the humoral response. I mentioned that already. That's the antibodies that are produced. On that last slide, it was the pink Y. The next part of the indirect measure is the cellular immunity, and we can measure that as a T cell response. Many of you may know T cells from the work that we do with HIV. We can check T cells and see how they're responding to our vaccine. The one I haven't mentioned is neutralizing antibody. And when you get vaccinated, you produce this humoral response that produces an antibody. We can measure the absolute amount, how much is circulating in your body. We can also actually measure how effective is that antibody in killing the virus. That's called neutralizing antibody. So in developing a vaccine, what the scientists and the folks in the lab do is they identify the antibodies, the humoral response, and then they measure in the laboratory the ability of that antibody to kill the virus. That's called neutralizing antibody. To move from the test tube or the laboratory to become a vaccine, an antigen has to have all three components. The direct measures of the effectiveness of a vaccine are really the one that's most important. It's the one that we've heard in the news this week. It's how many people get infected without and with the vaccine. So how well does it protect you? That's the question. That's a direct measure. There's two ways that can be done. Natural exposure is the ones that have been talked about over the last 2 weeks. The Moderna vaccine, the Pfizer vaccine, the AstraZeneca Oxford vaccine, those three all have been in the news over the last couple of weeks. They all did natural exposure. Some people got the vaccine, some people got a placebo. You let them out into the world and you see how often you get COVID. A challenge study, on the other hand, and there is a challenge study being done in England, there isn't one that I'm aware of being done in the US, implies that you give the vaccine to a certain group of individuals, you give a placebo to a certain group of individuals, and then you expose all the individuals to the germ. So in this case, to the virus that causes COVID, and see what the rate of infection is in those individuals who got the vaccine compared to those who got the placebo. Pretty incredible that you can get people to volunteer for that, but they did. It was done in England. The results haven't been published. There was a lot of discussion in the US prior to the results of these studies about whether we should pursue a challenge study. We didn't do it. But regardless, we measure, that's another way to get a direct measure of how effective the virus is. How about side effects? That's a very important question. It's one that's got a lot of people concerned because of the speed with which these vaccine trials have been done. And there's two principal things we're looking for. We're looking for early side effects and long-term side effects. Early side effects evolve over a 7-day period of time after the vaccine. They can either be local side effects or systemic side effects. Local means at the site of the injection. So a lot of us have gotten vaccines. You get your flu shot, you get tetanus shot. Where you got the vaccine or the shot, it hurts a little bit, it swells a little bit, it may get red. Those are the local effects. The systemic effects are how you feel. So when you've gotten a vaccine, you've gotten a flu shot, for example, maybe you feel a bit achy, your muscles hurt, you got some fever, some chills. Those are the systemic effects that you can have when you have a vaccine. Long-term side effects are things that occur over weeks to months after the vaccine. We are not going to have the benefit of understanding fully long-term side effects before the vaccine is given. At this particular point, we have 2, 3, 4 months of information on the vaccines. We don't have 2, 3, 4 years of information. And we probably don't have time to get that given the urgency of the pandemic. But what I can tell you about long-term side effects is that in general, because of the way that this vaccine, the two vaccines I'm going to spend the most time talking about, the Moderna and the Pfizer, were developed, it's not expected that we would have significant long-term side effects. Most of the long-term side effects that you hear about, Guillain-Barré, there's a long-term side effect complication for yellow fever. You can get yellow fever from it, from measles vaccine, from polio vaccine. Most of those long-term side effects come from vaccines that are made from the whole virus. The virus is attenuated or partially killed, doesn't work so well. And it's in that setting in which we tend to see more of the long-term side effects. There are a few whole virus vaccines being developed for this disease, but most are not. And so we don't expect a lot of long-term side effects. But again, we don't know that. So that is going to be a bit of an unknown as we all step up to get our vaccines. Now, when we are looking to vaccinate the population, our ultimate goal is what's called herd immunity. Herd immunity is here's the definition for you, but it's the indirect protection of susceptible individuals from an infection when a sufficient portion of the population is immune. It's important conceptually to know that there's a threshold for that. You need a certain number of people to be immune so that the entire population is protected. And that threshold is dependent on something called R, and we call that naught. It looks like R0 to you. The term for this is R naught. And what this represents is the number of secondary infections caused by a single infected individual in a completely susceptible population. So if I had COVID, how many people would I pass it to? That's R naught. And I'm going to show you a slide here in a minute to describe that. This is a visualization of herd immunity. And what this is really trying to show in this first panel, what you can see, all the folks in blue, they're a fully susceptible is what we call it.
I want to watch this.
And every one of these people could be infected. Here's Dr. Nahass sitting in the middle of this crowd, not doing anything he's supposed to be doing, and he's infected. As a result, since I'm not wearing my mask, I'm not speaking with a low voice, I'm facing everybody, and all these people surrounding me are doing the same. So we're at some gathering that's violating the law. None of us have masks on, and I'm infected in the middle of the crowd. All these people are susceptible. The herd, if you will, H-E-R-D, the herd, which is in blue, is fully susceptible. I can transmit this to a lot of people. No one has immunity. Second panel, a number of the individuals, they're in green, have become immune. There's some level of immunity in the herd, but they don't have full herd immunity because here I am in the middle of this crowd at this gathering that we're not supposed to be at. And even though there's a couple of folks that can block the virus from getting around, I nonetheless can still create a fairly broad and large epidemic, if you will, within this population. But we go to the third panel, and what you see is that a whole lot of people are now green, and they're kind of surrounding me. I have no way for the virus to get from me to the few people that are left that are susceptible. That's what herd immunity is. The herd, all these green people, get enough immunity that it protects the folks who are not immune. The only way to get long-lasting immunity in a population is through vaccination. And the reasons for that are very important. Herd immunity that occurs naturally, and there's some who would advocate this, can result in large number of deaths when the infectious disease is highly lethal. In 1918 to 1919, the United States and the world experienced actually our last true global pandemic. It was the Great Flu, called the Great Flu or the Spanish flu. Millions and millions, tens, 15, 20 million people passed as a result of it, a huge percentage of the population. And the reason was there was no vaccines back then. There was no way to stop it. The only way to stop it was to let the virus induce herd immunity. But that virus was highly lethal. And so in doing so, it did what it wants to do, which is go around, infect as many people and kill as many people as it can. That's the same risk with this particular virus. It is pretty lethal. Plenty of people survive. We all know that. We've known friends or family or coworkers who've had it. But an awfully large number of people succumb to this. In the United States now, we're knocking on the door of 300,000. And remember that number, because this slide shows you what percentage of the population needs to be immune in order to prevent the virus from spreading. For COVID, it's about 65%. The arrow's a little bit off, but it's about 60 to 70%, let's say. At the moment, in the United States, it's estimated that somewhere between 10 to 15%, probably closer to 12%, of the population's infected. So we can do the easy math. If you need 60% of the population to be affected by this virus to get herd immunity, and we've had 12% of the population already affected, we need 5 times as many people to get infected to achieve herd immunity. Well, 300,000 deaths, 5 times as many people get infected, 5 times 300,000, that's 1.5 million people. That's another 1.2 million deaths. Now you might say, yeah, but I hear the doctors are getting better. We are at managing the disease. I hear that the death rate's declining. It is, but not that much. It's declined a fair amount, but we still have plenty of people succumbing to this virus. It is a nasty virus. And frankly, it would not be an ideal way to achieve herd immunity to go to that 60% naturally. We would have a lot more death, and it would be very difficult for all of us to watch that. Okay, let's talk a little bit about about some aspects of the development of the vaccine, one of which is just the amazing speed with which it was developed. This slide illustrates the time frame over which other vaccines have been or frankly have not been developed. Just as an example, we now have an Ebola vaccine that's actually pretty effective, but it took 15 years. Polio vaccine, early days of vaccine understanding, took 60 years. We still don't have a vaccine for SARS-CoV-1. We still don't have a vaccine for MERS. SARS-CoV-2, the COVID virus, we went from knowing about the disease to having a vaccine that is now going to be deployed, probably starting next week, in less than 12 months. Really an extraordinary accomplishment. So why did that happen? Why were vaccines, why has the vaccine been so fast to develop, and how are we able to do that? Well, there are a number of reasons why. One, it was truly a huge global effort working with the world's leading scientists. It's really kind of a remarkable thing when you look around all the different places. You have all these major pharma companies that have an amazing bullpen, if you will, dugout of knowledge, scientific understanding, and resources to apply in almost a uniform approach to try and get this accomplished. There was really nearly unlimited resources, money, knowledge, manpower, technology, whether it be governments or pharma. It was a singular effort to fund it, both from a point of view of money, manpower, as well as manpower and technology. And then, frankly, the general population as a whole stepped up in a big way to volunteer and help by volunteering to be vaccinated in a placebo-controlled trial, all of which was necessary. I'm going to show you the size of these trials. It's really quite extraordinary to think about getting this many people involved in such a short time. So I mentioned earlier about some of the models of about how we do vaccines. I wanted to spend a minute just to give you a flavor of how far our science has advanced in our ability to develop various methods for vaccination. So there's principally two vaccine concepts that have been pursued in the development of the vaccine for SARS-CoV-2. One is what we call antigen immunization. And I spent a lot of time explaining antigens to you because I knew I was coming to this slide and I wanted to make sure you understood antigen concept. So we'll talk about this in a minute because it relates directly to the mRNA vaccine. And then there's virus immunization. Now, virus immunization, you already know. For example, the measles vaccine is an attenuated, partially killed, if you will, virus that is given to humans to induce immunity. That's what's called a whole virus immunization. Another way you can use virus immunization is to actually take inactive virus. So there can be an attenuated or partially alive virus, or you can take a killed inactive virus and use that to vaccinate the population. Both of these are what's called whole virus immunizations. They're being developed for SARS-CoV-2. They're nowhere near ready for prime time. Most scientists, most infectious disease doctors are not so keen on using virus immunizations. And frankly, these are probably going to just go away because of the remarkable success of the mRNA vaccine. So now we'll move up and talk about antigen immunization. And there's really two particular methods. Remember on that schematic that I showed you the virus and the pink Y antibody. There was a triangle on the outside of that virus. That was the antigen. That's what causes the response when it's given to us. You can make that antigen outside of the host, outside the human, in a test tube. There's technology to do that. The Novavax vaccine, which is being studied, Novavax is a company, I think they're in Delaware, it's an American company, and they make the antigen outside the human, they make it in a test tube, just like Flublok. The vaccine that we give for flu in this office is Flublok. Same concept. And then that antigen is given to the individual, just like you get Flublok here. You'll give this vaccine, the Novavax vaccine, and you'll give the antigen to the person. They'll make an antibody response, and they'll be vaccinated. The other way you can make the antigen is you can have the host, the human, me, make the antigen, make my cells a factory. How do we do that? This is the new technology. This is the new science. Very exciting. You have to deliver what's called the nucleic acid to the cell, which then allows the cell to become the factory. Nucleic acids are the genes. Our DNA is a bunch of nucleic acids. The genes of the SARS virus, the coronavirus, is a bunch of nucleic acids. If we can figure out what part of that nucleic acid is important to make the antigen for the virus, and we can deliver it to the cell, then we can get our cells to produce the antigen. Lo and behold, it works. And it's very exciting because the mRNA vaccine, which you've heard about, mRNA is the Moderna and the Pfizer vaccine, or viral vectors of the nucleic acid. So Moderna and Pfizer is a direct injection of the nucleic acid, the genes of the virus. You can use a viral vector to deliver the nucleic acid. Most commonly, it's a virus called adenovirus. That's the vaccine that you heard about in England, called the AstraZeneca Oxford vaccine. They've used a virus to deliver the nucleic acid of the SARS virus to the human. But all three methods, whether you inject DNA, RNA, or you use a viral vector, all three deliver the nucleic acid to the cell, then the human cell makes the antigen. Really cool science. It's the method that most closely mimics normal infection, if it can be normal, of the virus when it affects us. which is one of the reasons why we're also excited about this as a technological advance. Now, there are two viral vectors that are being actually very vigorously studied at the moment here in the US. And there's actually two that are already released and out in the, if you will, in various countries being used. And again, these viruses do not cause human illness, but they deliver the genetic material to human cells, allowing the cells to produce the antigen necessary to immunize. What are the examples? We can use human virus. It's an adenovirus. It has been altered so that it can't cause human illness. The J&J vaccine, or the Janssen vaccine trial, it's being widely done in New Jersey, uses this particular method of delivering nucleic acid to the cells. Also, the Chinese vaccine and the Russian vaccine that you've heard about that have already been deployed in China and in Russia use a viral vector vaccine, a viral vector to deliver the nucleic acid. These three are so-called human adenovirus vector delivery systems. You can also use an animal virus. And it turns out the one that you heard about this past week, the Oxford AstraZeneca vaccine, actually is a chimpanzee adenovirus. And they use this chimpanzee adenovirus to deliver the genetic material of the SARS-CoV-2 virus to the human cells. All of these do the same thing. They allow the cell to produce the antigen to immunize you. Well, here is the result of the AstraZeneca vaccine trial that was just released out of England this past week. It's very impressive. Let me just walk you through this graph. There's two panels that are of interest for you to look at. Panel number one, panel number two. It's actually a little bit backward in the sense that panel number two is the days since the first dose of the vaccine. Panel number one are the days since the second dose of the vaccine. So you got to go, if you will, right to left to understand the sequence. So if you look at it, the pink or orange color line represents the vaccine group. The blue line represents the placebo group. In this case, the placebo group was given a meningitis vaccine. And what you can see is the difference in the number of cases of COVID in the group that got the vaccine, the pink or orange line, versus the placebo. Low number, high number, and the difference between them at 150 days. So what is that, 5 months? Here is after the second dose, right? And the second dose comes a month later, so it's 100 days. What you can see is the difference between those who got the vaccine, the orange line, versus those who got the placebo. It's a bit bigger between here and here compared to here and here. In fact, what was the difference? Look over here, left side of the screen. The protection was 70.4% after 2 doses and 64.1% after 1 dose. So a little bit different, not much. But nonetheless, still very impressive in regard to the difference. So basically proving that the vaccine, when it delivers the nucleic acid to the cell, allows the cell to successfully produce an antigen, which will allow the human to become immune to infection. Again, very cool stuff. Here's the side effect chart. You don't have to look at every one of these panels. I'm going to help you walk through the slide. I think the thing that's most important is to recognize there's shot number one, shot number two. The yellow represents the number of people who have a side effect. And what can you see? You can see that there's less yellow after the second shot as compared to the first shot. Okay, we can accept that. What are the most common side effects? I have 2 panels to show you. These are the local effects: pain at the site of the vaccine, tenderness at the site of the vaccine. Again, as I said at the beginning, you've all gotten vaccinations. It would be a little bit surprising to get a vaccine and not have some discomfort and not have some tenderness. I think the point to note is that none of these side effects were either considered severe and none were overly severe, requiring hospitalization. So soreness at the site of the vaccine, tenderness at the site of the vaccine is what's expected. But, if you look at the systemic side effects, fatigue, fever, chills, basically looks the same. Again, second dose, less than the first dose, but nothing you wouldn't expect. When you get your flu shot, maybe the next day you feel a bit fatigued. Any of you have had the shingles shot? It's awful. My body ached forever. So those side effects are expected. So that's the virus vector vaccine. This is the antigen vaccine. Remember, I mentioned Novavax. Novavax produces the antigen in a test tube and then takes the antigen and gives it to humans. It's very similar to vaccines we already use. So this is kind of almost the oldest technology with a vaccine that's currently being studied. And these vaccines, examples of these that we currently use, this is Flublok, Shingrix, or the hepatitis B vaccines. All of those are antigen subunit vaccines. The antigen's made in a test tube and then administered to the human host. This is the result. This is a quick slide to just show you the effect of the antigen from the Novavax vaccine on humans. And what this tells us is this is the quantity of antibody. Remember, antibody is what's made. This is the dose of the vaccine. And what you can see is that when you give the vaccine, whether it be in the 5 or the 25 microgram dose, you exceed or equal to or higher than what would normally be expected from infection. This panel here is in normal infection. This is how much antibody is produced. When you give this vaccine in these 2 doses, this is how much you see. So for this reason, this vaccine is going to move forward, and hopefully we'll have some information in short order about how effective this vaccine is. Side effects at this point are similar to what I just described: tenderness at the injection site. In some cases, placebo was pretty common. I had to show you this slide. This is kind of interesting to me. I guess I didn't put it in there. But it's important to understand when we do these studies that we're always comparing to placebo. In this particular study, the side effect from placebo was almost half of the side effects seen from the vaccine. Why is that important? It's important because let's just say the frequency of side effects with the vaccine was 40%. And the frequency of side effect from the placebo was 20%. That really means that the vaccine itself is only creating an extra 20% of side effects or toxicity. So it's always important to look at these studies and compare the reaction to placebo so you understand the contribution of the vaccine over and above what might have been expected with a placebo. Now we're going to move on to the first 2 vaccines that are going to be available here in the US. And these are going to be, both are going to be mRNA vaccines. And there are 2. This takes advantage of that new technology that I mentioned to you, where we deliver the mRNA, the gene product of the virus, directly to the human. One is made by PFIZER and one is made by Moderna. Neither of them contain the COVID virus. That's really important. All they contain is a small, the term we use is a snippet of RNA. And that snippet of RNA codes for the spike protein. It allows the human, our cells, to make a harmless piece of protein called the spike protein. Remember that schematic, that diamond on the surface of the virus. And in doing that, it allows us to be protected, as we heard in the news. We'll show you that in a minute, sorry. That's really what it is. There is nothing else that RNA can produce. It can't produce a virus. It can't produce any other antigens. It only produces that spike protein. At this particular point, we actually have no commercially available mRNA vaccines. This would actually be the first commercially made mRNA vaccine. So Who was included in these vaccine studies with Pfizer and Moderna? And I had mentioned this to you a few minutes ago. It's really quite extraordinary. There were 45,000 people enrolled into the Pfizer study, and we're now over 30,000 people in the Moderna study. So imagine 70,000 people enrolled in a clinical trial over the space of 3 months, 4 months. Really an extraordinary accomplishment, and one of the reasons, frankly, they were able to get this done so quickly. People really stepped up to enroll. It's pretty diverse. I think this is an important point. Ages, a lot of people have asked this. It was great that they were able to get a significant number of people who were, above 50 or 60 or 70 or even 80 years old, because these are the groups that have the greatest risk for succumbing to this disease. How effective were these vaccines? Man, they are really good. It's amazingly good. And I'm going to show you more detail on the Pfizer success rate, frankly, because I have that information. It's been published. But the Moderna, it's not been published, but both really essentially at about 94, 95% protection. And it really had no difference regardless of race, ethnicity, or age. What about the side effect profiles? Well, this is the side effect profile for the Moderna vaccine. I'll show you the side effect profile for the Pfizer vaccine in a minute. This actually comes from the study that looked at the dosing range. For your purposes, focus on the 100 microgram dose, because that's the vaccine dose that'll be distributed across the country. And what you can see, here's vaccine 1, here's vaccine 2. Remember, Moderna's at day 1 and day 29, so it's 4 weeks apart. What you can see, vaccine 2 has a bit more side effects compared to vaccine 1. These bars are longer. What you can also notice, which is important to see and to understand, is that the severity of the reactions does increase from dose 1 or vaccine 1 to vaccine 2. But if you key in on the dosing, you'll notice that most of the more severe reactions were in the 250 microgram dose, not in the 100 microgram dose, which is the vaccine dose. So I really wanted to make sure that that was clear, because I do think that's important. If you look at the top panel, any symptom, what you can see is that really essentially almost all the severe reactions were limited in the 250 microgram dose. So side effects were manageable. Tylenol, heating pad if the pain is bothering you, but generally manageable with the Moderna vaccine. What about the Pfizer vaccine? Again, very dramatic results. This is what's called efficacy analysis, and this is looking at the results of COVID in the group that got the vaccine versus the group that got the placebo. And here it's broken down for you. This is the vaccine group. This is the placebo group. 18,000 people in both groups. Extraordinary, right? 3 months, 36,000 people. Gives me a headache thinking about it. What were the rates of disease in the two groups? Placebo group, 162 cases of COVID. Vaccine group, 8 cases of COVID. That's over a 2-month period of time. That's really unbelievably successful. When we do studies like this, we evaluate what's called a confidence interval so that we can say, how sure are we that these results are real? So you calculate a confidence interval, and given the size of the study, 36,000 people, the confidence intervals are narrow. So it goes from 90% to 98%. We are virtually certain that this vaccine will protect you at a minimum 90% and at a maximum 97.6%. And in the study, it was 95. So that's the confidence interval. That's how that's a really good vaccine. Very exciting to see. And I mentioned earlier about the ages. It's really important to be able to reassure older individuals. I guess I'm in that group. I am quite a bit older than 55. What we see here, again, 48 cases in that age group in the placebo, 3 cases in the vaccine group, and there were 7,500 in each of them. That's a lot of folks in that age group that were tested. So again, really remarkable results, 93.7% protection. What about the side effects? Here's the list. Again, it should be reassuring to all of us because the side effect profile was analyzed in 18,000 people who got the vaccine, 18,000 people who got the placebo. Here is the general disorders and administration site conditions. How often did you see a side effect? About 18.7% in the vaccine arm, 3.9% in the placebo arm. When you break it down, 11% for injection at the site of the vaccine. Boy, that doesn't seem all that out of the range of what I'd expect for a vaccine based on the vaccines I've gotten before. And then other things follow suit, 5% with fatigue, 6% with pyrexia as fever, chills 5%, muscle aches, joint aches 7%, headache 6%. So, all within a range that I think we should all feel awfully good about, that it's not overwhelming and certainly consistent with what we've seen with other vaccines we've gotten. Last thing, just to emphasize this, here is the same information for a vaccine we give all the time in this office, the shingles vaccine. If you look at the rate of side effects with the shingles vaccine, this panel is the local side effects. This panel are the systemic side effects. Look at the rates. 80%, 60%, 38% for pain, redness, depending on your age, a little bit more common if you're younger than older. What about fatigue? What about fever down here? Again, ranges that are not surprising to all of us because we give this vaccine. Some of you may have had it. But it points to the fact that having an adverse reaction, having a side effect from a vaccine is common. Doesn't matter which vaccine it is. And there's a reason for that. You get a vaccine to induce an immune response. An immune response causes symptoms. So having symptoms is not necessarily a bad thing. In fact, lots of times when a patient will complain to me about their symptoms, I'll say that means you had a vigorous immunologic response. It means you'll be protected from the vaccine. Got to get you through these next couple of days. But in and of itself, the response is not necessarily particularly surprising or worrisome. Certainly, if it's severe, we'd be worried about it. But that was not seen in the 40,000 people who've already gotten a COVID vaccine. So my final comments on safety, and then we'll move on durability. Last 4 months, many tens of thousands have received a vaccine. Life-threatening toxicity has not been seen. The side effects are on par with what we see with other vaccines, and it's important to understand that vaccine reactions are part of the immune response, so you should see them. The mRNA vaccines are only present, they only exist in our cells in the cytoplasm. The DNA vaccines, which have yet to get to large clinical trials, are not incorporated in our genome, in the genes, but they are brought into the nucleus. And there are some folks who have some concern about that. It is not incorporated, however, within our DNA. I should point out, as a point of interest, the DNA vaccines are far more stable than the RNA vaccines. The RNA vaccine has to be kept at -70 or -20. The DNA vaccine can be kept on the shelf. It's absolutely stable. You will not get COVID from these vaccines. And in regard to large-scale results from the other vaccines, they're still to be reported, so we don't have more information on them. Just emphasize this slide. The vaccine can't give you COVID. You just got to remember that. And you have to be comfortable with that. I'm comfortable with that. There's no reason to believe, there's no way that this mRNA vaccine can give you COVID. It can't happen. What about durability? That's a big question. How long will it last? At this particular point, the humoral or antibody responses for these vaccines or the virus appear to be present for 6 months or longer with the SARS-CoV-2 virus. So that's good news. We'd like it to be longer. We don't know yet. Not enough time has passed. However, we do know that the antibody responses with similar viruses, the SARS-CoV-1 and MERS, may be present for up to 3 years or maybe even a little longer. With any luck, we'll be in the same situation. 3 years would be like a home run in my mind for this particular virus and these vaccines. But we just won't know that till time passes. Same thing for the cellular response. It appears that it's going to be long-lived. We just don't know yet how long. My final comments on efficacy, how good these vaccines work. For the mRNA vaccine, we now have 2 different manufacturers with slightly different constructs, which have given us a proof of concept. This is really neat stuff, neat science, that this works and provides a high level of efficacy with a very acceptable level of toxicity, comparable to what we see with other vaccines. So far, the virus vector vaccines, the AstraZeneca Oxford vaccine, for example, does not appear quite as high as the mRNA vaccine. And that's true with the Chinese adenovirus vector vaccine and the Russian adenovirus vector vaccine, although I haven't seen the data in a formal publication. What's been sort of put out there would suggest they're not quite as high as the mRNA vaccine. Even one dose of both the virus vector vaccine and the mRNA vaccine provides some degree of protection. That's good news for all of us. I can't wait to get my first dose. We don't know the durability to an extended point, but right at the moment, it seems we're going to get certainly 6 months, maybe a year, and maybe, if we're lucky, 3 years. The biggest hurdle to safely reaching herd immunity is going to be vaccine hesitancy. I'm sure you have heard it. People are hesitant. They don't trust. I think that's in large part just part of the challenges we've had in dealing with this infection. We need to be part of the solution to this and encourage everybody that you see. You don't need to be hesitant. This process has been, I think, extraordinarily well done, and I'll speak to that in a minute. What about the distribution? You all have heard about this. The government's doing it. Certainly it leaves us pause, but they are trying to stratify it because you can't vaccinate 300 million people in 1 week. And so they're identifying so-called high-risk individuals for phase 1. This is the list. The first to be vaccinated is going to be this group. Second will be this group. Third will be this group. So it's going to go healthcare personnel, essential workers, adults with high risk, and adults greater than 65. That's the sequence of events that's expected. I think that will remain to be seen how that gets modified. But it's really out of the control of the general medical community. It's under the control of the federal government and the state government. This is what the schematic looks like in regard to sequencing it. Phase 1A, 1B, 1C. We're expecting phase 1A to start next week. So over the space of 5 to 6 weeks, all those in that first group I just showed you would expect to be vaccinated around the 4th or 5th week. So say early part of January, we expect to start vaccinating phase 1B. At least that's what the plan is. Phase 1C, somewhere around 3 months, say. And then everybody else, somewhere around 24 weeks, 6 months, right? So you've heard that in the news. That's kind of what the overall scheme looks like. I'm just going to run through these. These are kind of common questions that I've gotten as I've gone through about how do we know the vaccine is safe and effective? Why should we trust it? Is vaccine hesitancy? When and how long will I be protected? Will I need to wear a mask? What if I had COVID? And where should I go for more information? So first, why should I trust it? I got to tell you, I've been, as many of you know, I've been involved for a long time in clinical research. I know a lot of the folks that have been very intimately involved in dealing with the development. The scientists have really been straightforward. You have to tune out the talking heads, the news media, and the politicians. I think that the FDA, the NIH, the Advisory Committee on Immunization Practices, and the CDC are really doing an outstanding job, although not always well communicated, I'll give you that, to make sure that we're following the standards we need to follow to get this done. We need to do it quick, but they're not skipping steps. So I feel comfortable with that. And I just want to encourage you that you should be comfortable with that. There are a bunch of committees that look at this to help understand that this is the right thing to do and in the right approach. And so I think that we should be able to trust the vaccine. It is really not, the message may be managed by the media or the politicians, but the process is not. And I think you should feel comfortable with that. When and how long will I be protected? Don't really know the answer. Just spoke to that. Most of the vaccines are going to be 2 doses, 3 to 4 weeks apart. You actually get protection within 1 to 2 weeks, partial protection, 1 to 2 weeks after the first dose. That's a typo. Sorry about that. With really the 90% protection is 1 to 2 weeks after the second dose. We really won't know how long this is going to protect us until time passes, and we may or may not need additional doses for this virus vaccine. We just don't know yet. Will you still need to wear a mask? Yes, you do. Why is that? Because one of the other things we don't know is after you're vaccinated, although you'll be immune to getting future infection, we don't know yet that you will be immune to transmitting the infection. I don't think you would be able to do that, but we don't know the answer to that yet. So the purpose in wearing a mask is not protecting you. You've heard this, on the commercials. It's in protecting others, because we don't yet know enough if whether or not when you're immune, you can no longer transmit the virus. And so the mask prevents your secretions from contaminating somebody else on the chance that despite the vaccine, you could still transmit the virus. My gut says we probably don't, but I don't know that. So I'll be wearing a mask after I'm vaccinated. What if you already had COVID? What do you do? Well, you're protected for some time, but again, we're not sure how long. As a result of that, and as a result of the need to get people who are not immune, if you had COVID within the 90-day period before the vaccine is available to you, you will not be vaccinated. So we're telling anybody that's up for vaccine, say, for example, next week we start giving it in the hospitals to the COVID caregivers. If they had COVID in November, they will not get the vaccine. They've got to wait 90 days. But 90 days later, you will be offered a vaccine after that period of time. And what about further information? If you want to look, what sites are reliable? I think, as I've already probably alluded to more than I should, the media is not helping us. The media is not helping us with this. I think these sites are reliable, whether it be the CDC, the FDA, or the New Jersey government site. They'll keep you up to date on what the plans for distribution are, where they are with the understanding of the vaccine. I would stick to these sites. I'll just give you one example of why I think the media has hurt us and not helped from today's discussion, and some of you may have heard it, and it may answer a question or two. The vaccine committee, the advisory committee of the FDA, voted 17-4. 4 people said no to the advancement of the Pfizer vaccine. Now, why wouldn't it be unanimous, given what I just told you? At least you should ask that. I would ask that question. So I'm listening on the news as I'm hearing this, and it turns out those 4 individuals voted no because the information available for Pfizer's vaccine had limited data on children. Despite the fact that the vaccine works extraordinarily well in people who are 60, 70, or 80, the group most likely to die from this disease. But because it wasn't studied in children, they didn't think they should approve it. That's ludicrous. What the scientists and the vaccine manufacturers decided was that we should test it in the highest risk groups. They didn't test it in children. They're actually designing and proceeding with a children's vaccine version, and there will be a trial for children, but it wasn't designed to do that. And so to think that we should otherwise not approve it makes no sense to me. But the news media picked up on that. And the stories this morning, as I was seeing them flip through on my phone, is they're interviewing all these people, who, not all, the 4 people who said no. And they're highlighting the no votes, creating additional skepticism within the population. That is not a service to the population. It's not a service to those of us that are trying to help everybody understand it, because it's a misrepresentation of what happened. And so I'm constantly challenged by listening to the media. And that's why I'm advising you to think that if you want more information, go to these sites. I don't trust the representation that the media always makes. And so you have to be careful about it. I like their diagrams, though. Their diagrams are pretty good. So finally, how bright is the light? Well, I think the light's about 200 watts. I can't wait to get the vaccine. My arm is ready, and I'll be first in line. And I do think that the data is convincing, and I think the safety data, at least in the immediate term, is clear. And I think that the new technology and the method of development have really provided a lot of comfort for me that this vaccine is not going to cause long-term harm. And with that, I'll stop. And I guess we're doing questions, if any, in the chat, if I can figure that out. Duration between first and second vaccine. So for the Moderna vaccine, it's 4 weeks between doses. For the Pfizer vaccine, it's 3 weeks between doses. So if I go for my vaccine on the 21st, I think that's my scheduled date, and then I would go 3 weeks later to get my second dose. Will we be getting the vaccine? That's a really good question. I don't know where private physician offices fit in the scheme. We are not, the office staff is not in 1A because we don't directly care for large numbers of patients with COVID. Certainly our organization does, but we're not as if we were in the hospital taking care of COVID. So I don't actually know the answer to that question. We'll find out over time. My suspicion is that most of the physician offices will be rolled up in phase 2, but that hasn't been fully decided. So, well, thank you, everybody. Have a good weekend.